Hitting record. Okay, recording is starting. This week we have Sergio Pérez Limón from Rory Sawyer's lab at Penn State, and he's going to talk to us about his PhD research. So Sergio, take it away.
Yeah, thank you so much, Dr. Jim, for the invitation, and thank you so much everyone for being here. As Dr. Jim mentioned, I'm going to talk a little bit about my research, or the research that I've been developing while working on my PhD. And the name of the presentation is called "The MexMAGIC Population Defines Signals of Clinal Adaptation in Mexican Native Maize." I want to say that there's a preprint available of this work already. So if you guys want to go there more into deep into this talk, you can also go back to this preprint. Hope it gets published soon. And I would like to start with some takeaway messages. The first is that the Mexican native maize varieties are connect populations growing over continuous environmental gradients. And as such, to understand adaptation in these varieties is necessary to understand it on many varieties to many environments. And because of the nature of MAGIC populations, the MexMAGIC can overcome some of the major limitations of genotype environment associations in the study of the genetic architecture in clinal adaptation. Before I start or I go forwards with the genetics part of it, I want to show this conversation between Efrain Hernandez Xolocotzli, which was a Mexican botanist, very important in the obtention and the collection of all of these materials back in a couple of decades ago. And it's one of the main, also a takeaway idea from my presentation today. I'm gonna say this in Spanish and I try to translate it as best as possible in English. It says that, ¿cuál de estos maíces es más breve? Pregunté. Dijo el viejo, canoso, de piel arrugada y curtida. El amarillo es de 5 meses, el morado de 6 y el blanco de 7. ¿Y cuál rinde más? El amarillo poco, el morado un poco más y el blanco es mejor. Ja. ¿Y por qué no siembra puro blanco en lugar de esa revoltura? El viejo sonrió mostrando unos dientes cristalinos y pequeños como los granos del maíz reventador. Eso es lo que dijo, eso es lo que digo, mijo. Pero dígame, señor, ¿cómo van a venir las lluvias este año? Óigame, yo soy agrónomo, no adivino. ¿Ya ve? Solo Tata Dios sabe. Pero sembrando así, si llueve poco, levanto amarillo. Si llueve más, levanto más. Y si llueve bien, pues levanto un poco más de las 3 clases. Now, coming back to the genetics part of the presentation, we can start defining local adaptation as the superior performance of a local variety in its native environment versus a non-local variety. For instance, we can see that the green corn grows in the red environment and the purple corn grows in the blue environment, but we cannot assume that our variety source from a given location is necessarily locally adapted to that environment. We need to perform an experiment such as a reciprocal transfer experiment, where varieties sourced from different environments are evaluated side by side in common gardens in each of these locations. This will give us evidence of local adaptation in a set of discrete populations at discrete environments. For instance, here, we grow both the purple and the green corns in the red environment and the blue environment and assess a metric of their performance. And we can draw something like a reaction norm. And as we can see, the green corn outperforms purple in the red environment and the purple outperforms green in the blue environment. This is something, this change in performance of varieties across environments is described as genotype by environmental interaction, and that shape of a range-changing GEI underpins local adaptation. We can translate this into a practical example. So for instance, we want to assess if Palomero toluqueño, or PT, is adapted to its native environment, which is the Mexican highlands. PT is a Mexican native maize variety cultivated under low input conditions in the central Mexican highlands. And we compare it to B73, which is an elite inbred line from the Midwest of the USA, developed for intensive mechanized production under high input at the Corn Belt. And as in the previous example, we grow B73 and PT in both the highland and the lowland environment in a reciprocal transplant experiment, and we assess a metric of the performance in the shape of yield components, ear weight, and total kernel weight. And as we can see in the lowland environment, B73 outperforms PT in both performance metrics, whereas PT outperforms B73 in the highland environment. We observe rank changing GEI, and we can say, or we can conclude that PT is adapted to the Mexican highlands. Nevertheless, this experiment or this type of study of Mexican native maize have certain limitations. So for instance, PT is a native maize variety, and as Dr. Bellon mentioned previously, it cannot be considered a cash crop. It has other meanings, it has other values, other objectives to grow corn, not necessarily as a cash crop. For instance, in this picture, we can see the decorations inside of a church in the Mazahua region of San Pedro. This is in Mexico State. And we can see that these decorations are made with popcorn. PT is a popcorn, so this is some of the uses of PT in this region, and it's also hand cultivated, hand harvested, whereas B73 is a temperate elite inbred line developed for hybrid maize production, highly mechanized with other great inputs, and mainly as a cash crop. So it might be better to compare native variety and native variety, or inbred line by inbred line, not quite both at the same time. So also the experiment was limited to one native variety haplotype adapted to a single environment, that even though that it's important and it gives us very nice information, it can also limit our general understanding about local adaptation in Mexican maize. And this is because Mexican native varieties, instead of being discrete populations growing over discrete environments, can be understood as connected populations growing over continuous environmental gradients. For instance, in the rect panel, we can see a bunch of colored dots. Each of these color represents a different native racial group of the 59 endemic growing in Mexico. And each point represents a collection made by Conavio Biodiversity Institute in Mexico in 2011. As we can see, these dots, or the color of these dots, is not randomly distributed across the map of Mexico. So for instance, on the East Coast, we can see more of the pale yellowish dots. At the central Mexico, we can see more like green, pale green, more like vivid green. At the west, we can see more like orangey. At the north, we can see red, etc., etc., etc. So this is because the distribution is not random. All of these varieties, or the sustained association of a given native variety with its native locality results in adaptation. So the study of local adaptation in Mexican native maize requires understanding adaptation of many varieties to many environments, not only a single environment with a single haplotype. So can we get a better understanding of local adaptation in Mexican native maize with a multi-parent approach? And that's why we proposed, or we developed a MAGIC population with native Mexican maize. And MAGIC populations are powerful resources to study genetic and trait variation in a panel of native crop diversity. MAGIC stands for Multiparent Advanced Generation Intercross, and as we can see in the scheme, it funnels down the genomic contribution of several different founders into a single population of recombinant inbred lines. In comparison to biparental populations, they represent a greater genetic diversity. Because of all the crossing scheme or the crossing rounds, it reduces population structure present in the panel, founder panel itself. And you can simultaneously assess the effect of multiple alleles at a locus. But in the case of this research, and most importantly, if we have the source environment information of each of these founders, we can also make an assessment of the association between environment and the allelic effects that we are observing for certain trait. So the founders of the MexMAGIC population hereafter are sourced from different agroclimatic groups, and these are 8 different native maize varieties from these 4 major environmental groups, and 3 of them were represented by inbred derivatives, and 5 of them were represented by open pollination varieties accessions. And what the MAGIC population allows us to do is not only study, as in the example with B73 and PT, is not only to study adaptation at the extremes of the environmental distribution, but rather inspect what is happening inside this distribution with the different founders that we can sample from all over the environmental gradient. And instead of studying adaptation in the form, again, of discrete environments and discrete populations, we can study in the form of clines. So when selection is happening along environmental gradients in continuous, as is in Mexico, and continuous population as in native maize, we can it results in clines. So cline is the gradual phenotypic or allele frequency changes along a geographical or environmental gradient. So clinal variation due to selection is indicative of local adaptation. So it is important to disentangle the effect of selection from demographic history. One of the main advantages of MAGIC populations is the reduced population structure. So can we use the MAGIC population to describe the genetic basis of clinal variation in Mexican native maize? Can we use this reduced population structure to assess the role of adaptation and disentangled adaptation from the demographic history? And we can start with a case of study, the genetic architecture of early flowering in Mexican native maize. So early flowering, maize flowering time is a heritable complex strain that is driven by multiple small effect QTLs. In the literature are well-defined latitudinal and elevational clines for flowering time in maize. And for instance, in Ruben's paper of HPC1, it's also highlighted the importance of early flowering in highland adaptation early flowering for highland adaptation. So we can start asking first, is early flowering segregated in the founders of the MexMAGIC population? Can we use the MexMAGIC population to study the genetic architecture of early flowering in Mexican native maize? So first things first, is early flowering segregated in the MexMAGIC population? In this table, we can see the 8 founders of the MexMAGIC population that are sorted from top to bottom by elevation, and they are divided in 3 different elevational groups: a low elevation group in the red square, a mid elevation group in the purple rectangle, and the blue in the high elevation in the blue rectangle. And we investigated the flowering time of these varieties reported by Ruiz Corral in 1988. He performed a reciprocal transplant experiment in a low, mid, and high elevation sites in Mexico. And the values reported here are the average the days to flowering for all of these varieties across the reciprocal transplant experiment. And we can see here highlighted in yellow are varieties that are segregating early flowering across this environment. So early flowering is in Zapalote Chico, here in red, Gordo, Palomero Toluqueño, or PT here in blue. So there are early flowering in both highland and lowland elevational groups. But also we have other varieties that are more late flowering, or late flowering is also segregating along elevational groups. So for instance, Reventador here in low, we have Jala or Mushito here that tend to have longer flowering times than others. So to perform or to assess the flowering time in the MexMAGIC population, we perform a common garden experiment in a mid-elevation site in West Mexico, in Ameca. So we evaluated 200 MexMAGIC families, test crossed by NC358, and we evaluated for 2 years in Ameca, and we recorded flowering time for all of these populations. To dissect the genetic architecture of flowering time, or early flowering time in the MexMAGIC population, we use an 8 allele QTL model, and we identify the top 1% SNPs and merge them into 11 different QTLs. So when we try to identify the source, the source allele for the earliest allele at each of these QTLs, we can see that there is not like a clear trend. It's not that there are a lot of accumulation of early alleles from any single parent. But when we go forwards and instead of using the alleles themselves, not the alleles, but the elevation, so the source elevation of each of these earliest allele, we can see that we can start observing a trend into the QTL. So for instance, here in chromosome 8 and this chromosome 4, the source of earliest allele comes from a highland parent, whereas in chromosome 2 and chromosome 9, the source of earliest or acceleration in flowering comes from a lowland founder. Drink some water. Now, when we assess the effect of 8 different founders across the 11 QTLs that we identify, we have a figure like this. So this is a heat map that represents or summarizes the allelic effect of the 8 founders at the 11 QTLs detected in the analysis. In the x-axis, the founders are sorted from low to high elevation. In the y-axis, we have the 11 different QTLs detected in the analysis, and the color of each square represents the allelic effect of each of the founders. Red alleles, in this case positive effects, represents early flowering, where blue alleles or negative effects represents delay in flowering. Now, in the y-axis, the QTLs are grouped or were clustered according to the allelic effect of the founders into 3 different groups. In A, we can see a group or a cluster or founders whose earliest allele or acceleration in flowering effect comes from highland founders from Gordon, Muschito, and Palomero. And as observed in the previous Manhattan plot, it comes from they are in chromosome 8, 8.2, 8.1, and chromosome 4. This B cluster, it's our QTLs whose source of early flowering are either mid elevation to low elevation. And this in chromosome C, we can see that the flowering or the early flowering comes from low elevation founders in Reventador and Alter Zaparote, something that we also observe in the Manhattan plot. So we can ask ourselves, is there any evidence of selection along altitude happening at the QTL level? Given that we could cluster this by elevation, can we assess evidence of selection acting along this altitudinal climb? And for this, we perform genotype environmental associations that use statistical association between allele frequency and environmental parameters at the location of origin to identify alleles adapted to local environments. So when we see, for instance, an increase in allele frequency with an environmental gradient, we can assess evidence of selection happening along this gradient. The recovery in the case of the QTL that we're observing, the recovery of the clinal pattern at the QTL level is consistent with directional selection acting on the QTL. And this is after the correction for population structure. And this is something that is called reverse ecological genetics, when we find a variant associated with adaptive traits and then test its ecological importance using GEA methodologies. When we apply a GEA model onto these 11 QTLs, we can see that we observe elevational climbs at QDT 8.1 and 8.2, and positive elevational climbs at QDT 2.3. This means that the latest flowering alleles at QDTA 0.3 comes from, again, comes from highland elevation, and that the recovery of the positive climb after correction for population structure means that there is selection happening at the QTL level. On the other hand, you can see that at 4.2 4.2, we don't see a very clear trend, elevational trend. So the line is almost parallel. But at 8.2, we can see that the earliness of flowering time comes from high elevation founders even after correction for population structure. So we can assess. So yeah, this suggests that the genetic architecture for highland and lowland early flowering is different, and it's happening at the QTL level across an altitudinal climb. So we assess evidence of this at the QTL level, but is this trend also reflected at the genotype or at the founder level? And when we perform the QTL sign test, so the allelic effects that we observed previously in the heat map were categorized as positive, negative, or neutral, and some across the 11 QTL for each of the founders. So positive allele counts would mean that they're early flowering, and negative that they are late flowering. So there is no significant relationship between QTL sign zoom with elevation. When we try to correlate the source of founder elevation against the QTL count, but this means that there is no significant evidence of selection happening at the genotype level, something that is not reflected at the QTL level. And as we can see, this is because at different elevation groups, we have families that can be very early flowering, or they can be very late flowering. So for instance, here, Reventador it's very late flowering, or the accumulation of QTL sign indicates that it's late flowering. And in this case, Mushito at the high level is also late flowering, something that is observed also at the reported flowering times by Ruiz Corral. So again, it's that because we are observing, we are looking at something that might not be the correct line. And when we assess another type of climb, for instance, this is a physiological climbing thermal requirement. Thermal requirement is the specific accumulated heat above a base temperature necessary for the crop to reach flowering. So when we assess now the QTL sign count against thermal requirement, we can observe a negative relationship between thermal requirement and the sum QTL sign. And this is a recovery of the climb that we were observing. So this means that across a thermal requirement, so that people even across different altitudinal groups were selecting or are selecting for early flowering or late flowering, and this is also reflected at the sum of the QTL sign. We recover the climb along a thermal requirement, which in turn might indicate that the selection is not happening at all elevation, but it's happening according to the thermal requirement. So this highlights the importance of farmer selection for early flowering at every elevational group. So as some kind of summary from the results is that this genetic architecture of DTA is polygenic with several QTLs segregating in the genome. QTL in chromosome 8.2, which is associated with highland adaptation, colocalized with loci associated to adaptation to high altitude and also to high latitude, in this case, CCN8, BGT1. Highland allele accelerates flowering in these cases. And the significant GA suggests selection acting along an elevation climb of this QTL. The clustering of these QTLs affect and the evidence of GA along altitude suggests that this genetic architecture is different across lowland and highland elevational founders, and that the early flowering is driven not, at least in this population, it is not driven exactly by altitude, but by thermal requirement that people might have selected for early varieties at every different group, and also for late varieties at every different elevational groups. Something that it's also mentioned in the talk with Efrain X at the beginning. And in this case, I wanted to compare, for instance, the effect or the advantages that the MexMAGIC population had along popular or against traditional GAs. So GAs uses diversity panels to assess or to obtain evidence of local adaptation happening along environmental gradients. GA assumes adaptation to a single environmental gradient, that this adaptation involves monotonic clinal patterns in allele frequency at the loci under selection. And it's used a lot because you only require data on the environment of origin and sequence data to assess this kind of evidence. But the limitations of this approach is that what if the evolution of local adaptation does not involve clines in frequency at the selected alleles? Or what if, because of population structure, we undercorrect or overcorrect for it, and it might lead to a failure to accurately detect the loci under selection. And in the end, the variants that we identify that are locally adapted or that are associated to adaptation to a certain environmental cline, we still need to validate its adaptive phenotypic effect on the population. In the case of the MexMAGIC population, even though that there not might be a clinal adaptation happening, we are still capable of dissecting the genetic architecture of adaptive traits regardless of these clinal patterns of adaptation. So we don't require, it's something more akin to GWAS. So we don't require the evolution of clines to really observe or to really understand the genetic architecture of these adaptive traits in comparison to GEA. Also, with related to the under or over correction for a population structure, the reduced population structure in the MexMAGIC population allows for the identification of clinal adaptation as well. So we have a large statistical power to detect these clinal patterns. And if we use a reverse ecological genetics framework where we already evaluated or we already know putative adaptive traits, we can use the reverse ecological genetics to see or to test the ecological advantage or the ecological importance of these variants across different environments. So we go from the phenotype or adaptive phenotype into the ecological and environmental importance and not the other way around. So we can get a better understanding of local adaptation in Mexican native maize with a multi-parent approach. Yeah, we evaluated several different, so we evaluated several different families in a single environment, but we still need to evaluate in different environments. So from the perspectives of this work is that we can increase the number of families. I am from 200 to 500. And it would be really, really cool if we could assess or we can perform a common garden experiments of the native environments of this population across all the native environments of the MexMAGIC population. Right now, we have data for mid elevation and high elevation sites, and it's something that I'm going to keep working on. This is a very important, something that I am very happy about and excited about is that I will join, well, if everything goes right, I will join Dr. Joe Gage lab at NC State as a postdoc during the summer. And we're going to perform, we're going to perform an experiment there where we're going to evaluate the genetic architecture of early flowering in two real populations, B73 by Zapalote Chico, which is one of the lowland varieties whose allelic effect, the chromosome 2 were the highest, and B73 by Palomero toluqueño, which is a highland variety and which early alleles come from chromosome 8. And we try to understand the differences, not only in the genetic architecture, but the regulation of early flowering between these two types of early flowering in this population. And I'm very excited to join the people at North Carolina State University, and also be in a warmer place because this winter has been awful. And just jumping into the acknowledgement section, thank you to former and recent lab members here in at Penn State, also in Langevio in Mexico, to Ana Laura, to all the people in Simbe staff. And I would like to thank to all the people that have been working with the different bits of the MAGIC populations at Iowa State University, Cortiva, MesGDB, Simbe staff, the different professors and people at NSF working on the Hilo project, and NC State, Dr. Peng Yu and Xiao Fang, and Technical University of Munich, to Wei Jun and Ed Buckler at Cornell. And also, and probably not least important, I would like to acknowledge the smallholder farmers and indigenous people of Mexico and the Americas whose work and love for their traditions and identity keep maize diversity alive. So, well, that's it from my side, and thank you so much.
Okay, great. Thank you, Sergio. Great. I put a couple questions in the chat, but maybe John also has a few, so I'm going to turn it over to John first and let him go.
Hey, can you hear me?
Yes, I can hear you.
Awesome. Really interesting talk. I love the scale of these types of experiments. And I feel like whenever I sort of think about the things that you can do with these magic populations, it always kind of gives me a lot of ideas as like an evolutionary biologist. I guess I was kind of wondering, and this was spinning off of Jim's first question, if you kind of know what the underlying genetics are within these rills, and you were sort of alluding that these subpopulations can occupy very broad regions of Mexico and Central America. Uh, are Do you have any sort of sense on if these alleles could be sort of across the whole subpopulation? Could they be locally adapted? And if you are sort of getting local adaptation, could you then sort of like look at similar clines across the edge of the population and start to look for, convergently adapted alleles? So basically kind of capturing the signatures mirroring each other for like they're achieving the same trait, but maybe through slightly different means.
Sorry, can you repeat the questions, please?
Oh, yeah. Sorry. Yeah, here, I'll feel like everybody else is also turning their camera on. I'll be polite. So I guess I'm just if you kind of have like a very broad range to this population, I guess I'm wondering like, do you have a sense if some of these alleles could be sort of like locally adapted to just this specific cline, or could it sort of occur across the entire like sort of range of where that sort of uh subpopulation type is? Because if it's sort of the former, then in theory, if you sort of like look at two different clients at the far edges of the range, you might have some sort of vehicle for asking how some of these traits like flowering time or other things could be convergently evolving. Um, uh,
Uh,
yeah.
so yeah. So answering your question, in this case, yeah, elevation, temperature, thermal requirements are like single variable clients. Where in reality probably they are more like multivariable clients in the sense that, for instance, elevation, at least in central Mexico or in Mexico in general, captures a lot of different environmental variations, such as temperature, such as UV radiation, even soil type, because all of these like the high plateau and all of these volcanoes in Mexico come from volcanic soil, so they have a even different difference of phosphorus retention, humidity, etc., etc. So in this case, probably thinking about a single variable environmental climate, it's a little bit reductionistic. Probably in these terms, the adaptation happens to multivariable clients, and sometimes even the multivariable client might not be detected by this EGA or other type of associations, because they might not even evolve clients at the allele level, which is something that is discussed by Lothero's paper. So yeah, it's a little bit. I wouldn't say again. I wouldn't say that it's reductionistic, or I wouldn't say that it's not relevant. But at the same time, it's important to think about all of these varied clients and how and it's something that it's also worth thinking how can we assess them using this type of populations. And I think coming back to your second question, some of these, at least in flowering time, there are several ways that you can be early flowering. So for instance, you have all of these in QTL, in chromosome 8, they are associated with temperate adaptation or to adaptation to latitudinal. Here, for instance, to the latitudes in Pennsylvania, other latitudes even northern from here. But for instance, Palomero toluqueño, even though that it has early flowering in the chromosome 8, empirically, we have observed that it's also photoperiod sensitive. Zapalote chico is not photoperiod sensitive. It has been reported as insensitive to the photoperiod, and it's also it's also getting to early flowering time. In Arabidopsis, I think I'm reading a little bit more about this nowadays, but there are different ways that you can have variation in flowering time. You have like the autonomous pathway, the photoperiod pathway, the gibberellin pathway, temperate pathway. So probably there are some pathways that are being affected by the direct selection of farmers and others that might be selected by natural selection, depending on the environmental conditions that you're observing. And it's something that I might try to assess with the experiment that I was mentioning previously with the B73 by PT and B73 by Zapalote Chico populations.
Awesome. That definitely kind of covers the general. Thank you so much. Yeah.
Thank you for your question.
Yeah, Sergio, I had a couple of technical questions. One is that the original parents used, were they individual, were they single plants in the first crosses, and were they outbreds, or did you have like inbreds selfed out of the populations?
Yeah, so the first these founders, the Sapchico, Tabloncillo, and Reventador, I think these are the MR accessions. They were developed by.
So they're inbreds.
Yeah, they're S6. They're not quite inbreds, but they're inbreds, yeah. And the other ones are open pollination varieties. I didn't mention it here, but because of the crossing scheme, we're only selecting one individual F1 plant, so we can capture only a single haplotype from the open pollination varieties that is inherited into the population.
But that definitely simplifies the technical side of things in terms of assigning haplotypes to founders.
Yeah, and even that is sometimes it's complicated.
Right. I had another question about your conclusions. It seemed to me like you gave two almost contradictory conclusions about 2 slides away from each other. You said that you're showing evidence that there's farmer selection for early flowering at each elevation. And then you said, if I understood it correctly, I think you said then essentially there's evidence for farmer selection, for kind of divergent selection for different flowering times at common elevation groups.
Yeah.
So I was just trying to, could you kind of just unpick that a little bit and clarify what your conclusion is?
Yeah, so sorry about that. Probably I might have been or I didn't say it properly, but at least with what we observe at the genome level or the genotype level here in the MexMAGIC population is that there is evidence of selection for early flowering and different types of selection for early flowering depending on at the QTL level. But at the genotype level, it's here. We also observe selection for early flowering in the MexMAGIC population. So again, the MexMAGIC population is only a small fraction of the overall diversity that we can observe out there in terms of diversity of corn. With this population, we didn't assess evidence for a late flowering in other families. or in other varieties. But we observe that at different, I mean, if you go out there, we observe that at every elevational group, even within the same farmers, you have early, average, and late varieties. It doesn't mean that it has been selected, or the conclusions that I can reach with this study is that I don't know if it was for selection, but at least what we observe in the MexMAGIC population is that early flowering in the founders is driven by farmer selection, and it's part of well, another like technical questions that we think about in the MexMAGIC population, that how much of the genetic and also phenotypic variation can we capture with this population, and how much can we extrapolate it to a greater diversity. It's something that I was
Perhaps,
working with.
can I jump in as well, Sergio, for that
yeah.
Jim's question? I mean, I think, Jim, the point is not that people are only selecting for precocious varieties at any particular elevation. Kind of going back to Sergio's original quotes, no, you're actively maintaining different maturity groups at different elevations, and potentially what we're revealing is through different genetic architectures. So it's not you only want early flowering, you want early, medium, and late flowering wherever you are. And so if you want, you could call that divergent selection, or as I say, just something different.
Make a couple slides later. It might confuse people though if you give them both of these conclusions, because they, at least on the face of it, they seem a little bit contradictory.
Right. So it's not selection for early flowering at the expense of any other maintaining anything else. You're maintaining different maturity groups at different elevations.
Yeah. Rory and Sergio, I totally, and I think, Sergio, your quote from the farmer right at the beginning, I think it was great, because I have no doubt that is exactly what they're doing, is they're maintaining a wide range of maturity in the population. What I don't know is, are they labeling the maturity groups with kernel traits? Okay. So, I mean, if you want to, because once things have flowered and the seeds all, and you put the wrong ear in the wrong place, and you're not controlling pollen and everything else, unless it's kind of labeled with a kernel color or a cob morphology or something, how do you know which is which? And that's what I was kind of wondering. Have you estimated FST? For kernel traits versus flowering time traits among these three different groups, class of genes and things.
That's a very interesting and good question, and it was something that I was also thinking about and discussing with Dr. Rory sometimes, like what could be like the different way to achieve certain phenotypes, right? In this case, I don't know. Answering your question at this point, I don't know, but it would be interesting to see, for instance, what are the ways to reach, for instance, early flowering, or the different mechanisms to reach early flowering, to the mechanisms to reach palomero type of kernel, or tortillas, or very specific pozoleros type of kernels. For instance, zapalote chico in the literature, and when you talk with people that work directly with maize diversity in Mexico, they call it cuarentano. Cuarentano means to 40 days. So in the native environment in the valley in Oaxaca and El Istmo, it reaches flower in 40 days, and the kernel type or the use of these kernels are very specific for totopos, which is some kind of have you ever had tostadas? It's like not it's like a baked tortilla. So I think that it will depend a lot also in the variety and the uses of the variety itself. But it's a good question.
Yeah. I remember when I was doing archaeology, now we're talking 35 years ago, I do remember a paper in Latin American antiquity that actually linked the two of them together. But be hard. And Jeff says there's evidence also of it, but I didn't know how you.
I mean, I think I've seen a few of those reports as well, Ed, but also obviously as you start to diverge flowering time, that itself will allow you to maintain distinct populations, if you like, in effectively sympatric cultivation. So that also can be useful to maintain these other more derived kernel traits and all the rest of it.
But it doesn't take too much gene flow to keep things moving, you know. So I mean, that's where I think with if you have 200 or 300 genes controlling flowering time across central Mexico, you can definitely get your means roughly there, but any individual gene could be all over the place.
Jeff had a comment. I don't know if you want to say anything, Jeff.
No, just following up on Ed's comment that I'm aware, at least in ethnographic literature, of color being linked to flowering time, at least locally. But to Ed's point, I would be surprised if I'm pretty confident that, for example, blue isn't genetically linked to any particular flowering time at a continent scale, even if it is within a particular village or set of villages.
Yeah, and Sergio, it'll be sort of interesting to see if you grow stuff here in Raleigh, what you'll see, because what you said about photoperiod sensitivity is, at least in Zapchico, that is my observation, is the populations I've looked at are reasonably early here. And it's the only thing out of Mexico I've ever seen like that. Everything else I've grown from Mexico has been super late, clearly photoperiod sensitive. I don't know, but as you said, it's also this 40-day. It also is a really per se early. So how much of that is due to, does it really lack photoperiod sensitivity, or is it just so early that overcomes, everything else? I guess you'll see. And then I haven't grown Palomero toluqueño here, so I don't know, but probably it's photoperiod sensitive. So you may see that, you may pick up some of that difference here that you wouldn't see probably in Mexico, or at least you'd see it much less in Mexico. It's my guess.
Yeah, you're right. Yeah, that's why I asked Ruben to see if we can grow some of the tester a little bit earlier, because the Sad Chico population, I expect it to flower like quite early. I don't know how early though, but probably earlier than the tester.
Yeah. Okay, any other questions for Sergio? Sergio, thanks a lot. Good luck defending your dissertation. The people here in Raleigh, I'm looking forward to your arrival here. So we'll thank Sergio one more time. Next week, I believe we have Ana Butron from Galicia will be presenting next week. Thanks, everybody. Thanks, Sergio.
Thank you, everyone.